Hello everyone, welcome back to this ultimate guide for access to HE. Today we will be doing lesson 2 covering specialized cells, and that forms part of the human physiology spec for introduction to cell biology. The criteria really wants you to relate structure to function in two specialized eukaryotic cells, and we come across the relationship between structure and function quite often this year. So as part of your first unit, it's really important that you get a good grasp on not only the knowledge required to relate structure to function, but also how you get that down on paper or on screen so that you can get those distinctions that you so deservedly want. So the term generalized cell refers to the things that we've been learning about recently. When we learn about a cell, we'll learn about the common components of cells. And we've been talking about the cell membrane, and we've been talking about organelles, and we've been talking about the proteins in and around the cell. But that's a generalized cell, and in reality, the generalized cell doesn't really exist, because all cells specialize to be or to do a particular job. So we know that each cell, or each of our somatic cells, i.e. our body cells, has 46 chromosomes inside each nucleus. Not all of those chromosomes are required, because at some point in time each cell will be told what their job will be, and only certain chromosomes have the information that that cell needs to be that special job. So if a brain cell, if a neuron, let's say, is told that it's going to be a neuron, then it doesn't need the instructions on how to make insulin, for example. That's the job of a pancreas cell. So what I'm trying to get at is that not all of the DNA and the chromosomes are required to be each cell. It only needs a fraction of that DNA, but it's enough DNA to give it the instructions that it needs to become that cell. So let's say a cardiomyocyte, a heart muscle cell, will only need a certain amount of information from the chromosomes to be a specialized cell. Now there's going to be massive differences between a neuron or a nerve and a cardiomyocyte, because this has to contract and it has to pump blood around the body and create force, and the neuron has to send electrochemical messages to and from a particular place, and you don't need any contraction there. So actually, when we look at the structure of a specialized cell, or when we look at the structure of any cell that has a specialized job, you'll see that they're built in completely different ways. The generalized cell is there for us to learn about, to familiarize ourselves with the common components of cells, so that we can identify how certain cells specialize, and that's what this part of the assignment is all about. So what I'm going to do, I'm going to run through a couple of terms, and then I'm going to go straight into it and explain 4 specialized cells, but I'm also going to give you hints about past merits and distinctions, what the kind of standard is for a past merit and distinction. And it's an ungraded unit, this introduction to cell biology unit, so you don't need to worry about getting this distinction, but it's a really good chance to push yourself to get the best grade that you can, so that you know what standard you're working at, and we can help you to develop your areas of weakness, and we can also build on your strengths. So it's really important that you give it your best shot, and I will be in this video giving you the simple version, and I'll also be giving you the advanced version as an example for you to take away and apply it to the ones that you choose. So it's really important to remember that a cell's structure reflects its function, which means that the way it's built reflects the job it does. So you look at anything that's been designed around you, your phone, your TV, your keyboard, the mouse. It's very easy to sort of look at how something is built and how it suits its function. I'm looking at my computer mouse right now, and you can see that it's a shiny material which feels good in the hand, and it's good for movement around. It's got multiple buttons on. It's at the right shape for either right or left hand, and there's a mouse wheel there. But what we have to do is first of all name its job, and its job really is to be the interface between somebody's hand and the computer screen. So it needs to be tactile, and it is. It's light, and it's of the right shape that we can grip it in the middle where it goes indents, and we can, within one or two movements of our fingers, we can have a left click, a right click, and we can roll the mouse up and down. So it has a very great amount of design features that allow it to be good at its job. So we wouldn't want a house brick, for example, as a mouse, because its design is not suitable for its function, i.e. its structure is not suitable for its function. So it's really important that you focus on the fact that, or ask yourself the question, why is a muscle cell built like that? Why is a nerve cell built like that? What job does it do, and how does it suit that job? And that's really the essence of this part of the assignment. So I've given you some examples there on the board or on the screen of specific cells that you could choose. I'm going to cover a spermatocyte, a erythrocyte, and a goblet and ciliated cell of the trachea. So those are what I'm going to choose for today's PowerPoint or video. You can look into a nerve cell or a skeletal muscle if you'd like. Some are far more complicated than others. You're only going to have a couple of hundred words to write in your assignment, so we're really after quality information and information that you're going to be able to put across about the structure and function of specific cells. So the question is, what job does that cell do? How is it suited to that job? And we're linking the two together. So let's walk into it. So here we've got a spermatocyte or a sperm cell, and really we're talking, if you're going to address this in your assignment, I really kind of express what the major role of the cell is, and it's fairly obvious that the intention or the function of a sperm cell is to pass on the genetic information from the father in order to fertilize the egg or an egg with the other genetic information. So if you've done the work on mitosis and meiosis, then we know that in humans there is 23 chromosomes in that nucleus which needs to fertilize or mix together with another 23 chromosomes from the egg to make 46. So that's where I would start with if I was going to choose a spermatocyte or any other specialized cell. When I'm talking about it in my assignment, I would give the overall function. Give the overall function of the cell, and that will provide a nice introduction to your bit of writing. Once you've got the overall function, then you can start looking down at the describing the structure. Now you've got really two options when you're writing an assignment on it. You could actually relate the structure to function as you go. So if you're talking about a particular structure or feature, you can then link it to the function. Or you can describe the structure in detail and then link it back to the function at the end. So if I describe the structure first and then I'll link it back to function, really what we've got here is three sections. We've got this section here, this section here, number 2, and this section here, number 3. So it can almost be split, the sperm cell, into 3 sections. The first section at the bottom is a flagellum, or flagellum, depends where you're from. And then in the mid piece we have mitochondria. And in the head you can see that we've got the nucleus and acrosome and centrioles. So that's a description of the structure. And what we can do then is link those structures to the overall function. And a sperm cell is one of the most obvious cells in order to be able to relate structure to function. So we can see that the flagellum is a whip-like tail, and it thrashes from around from sort of side to side, which is used for locomotion, which is a posh term for movement. So that's obviously important in order for the sperm to get to its destination. The midpiece is crammed full of mitochondria, and if you've done the work on the generalized cell, then we know that mitochondria are used for energy production, specifically ATP. And obviously, I would say obviously, the energy is there, a lot of it is for locomotion, so it's to provide the energy for the fuel for the sperm cell to go on its marathon swim, if you like. And then finally, what we've got in the end here is we've got an acrosome cap, which is on the end, and we've got the nucleus inside the head, which makes up most of the head. As the sperm cell gets to the egg, and I'd like you to learn about this as much as you can, the acrosome then releases some enzymes which break down the cell wall. Sorry, not the cell wall. The wall of the egg, or the oocyte, which is the correct term for an egg. And so that acrosome then allows the nucleus to sort of fertilize the egg. And so, but once that's happened, then once a sperm has entered or been allowed to fertilize the egg, then the egg goes into lockdown to stop polyspermy, which means that obviously there's millions of sperm going on search for this holy grail, and so it stops multiple sperm from fertilizing the egg. So that's the acrosome. It's an enzyme which breaks down the outer lining of an oocyte. And finally, obviously, we said it at the start, the nucleus has 23 chromosomes in, which are there to fertilize the egg, to pass on that haploid number of chromosomes and make the first full diploid number of a new human species, or any other animal if you like, but just with a different number. So that's the kind of thing that I would put in terms of a spermatocyte, and I would be looking to use some sources to support what I've just said there. So it's down to you as a bit of a research task to support what's been said. The second cell that I'll talk about is an erythrocyte, which is a red blood cell, as you can see. In terms of the function, it's there to carry oxygenated blood, oxygen around the body for respiration. So it's a delivery cell which delivers oxygen to the other cells or other tissues so that they can use it for respiration. So what structural features make this excellent at obtaining oxygen and also delivering it well? You can see that the cross section at the top here, if I were to draw a sort of disc shape cross section, you'd see that was very flat. You'd have two flat edges, one at the top, one at the bottom. So you can see this, it's called a biconcave disc. A biconcave disc shape, and that increases surface area so that it is a much more efficient exchange surface. Right? I see a lot of students saying that it can carry more oxygen. It's kind of ambiguous, because actually if it was a big sort of solid shape, or even if it was a big blob, it would be able to carry more oxygen inside it. It's not about carrying more oxygen per hemoglobin. It actually increases the exchange rate. Don't forget it carries carbon dioxide back as well, but it increases the speed at which it can absorb oxygen into it. and therefore if you sort of take the blood as a whole, you could argue that it carries more oxygen because it takes it up at a faster rate. But really what people confuse it with is that it can carry more oxygen per red blood cell, and that's not quite the case. It increases the rate because it has a bigger surface area than if it was just sort of a normal flat disc shape. The fact that it's biconcave, i.e. there's a dimp there and dimple there, is that it increases the surface area. So if we measured around this disc here, let's say it's 10 centimeters, whereas if you measure around this disc it might be 12, 14, 15 centimeters, and so that bigger surface area allows us to soak more in, if you like, as it passes through the lungs and the alveoli. There is arguments, and whether you can find the research into it, that because it's got a disc shape, when it's being pumped, because it's fluid, and as it's going around the blood vessels, this biconcave disc allows it to be, pushed through the system better. That's something that you might well find. Another feature of a red blood cell is made of a protein called spectrin, which allows it to sort of change shape a little bit, to squeeze, and if it's going through tight spaces, it'll allow the red blood cell to change shape a little bit and get through tight spaces, and that will help the flow of blood through it. We can also say that it's full of hemoglobin, and hemoglobin is an essential protein, which is the thing that oxygen binds to. I'm going to challenge you at this point to find out a little bit about hemoglobin and why it's so good at holding on and giving away oxygen, and now we can go into different levels of that. I don't expect you to go into full detail, but that will embellish your assignment to more than I'm saying here. But the two real key features are, or the main feature is that it's a biconcave disc shape, which allows a faster exchange of gases because of the increased surface area, but also it's made of spectrin, and it's also full of hemoglobin. So take that where you will in terms of structure and function, but both of those, or all of those features, help the function of carrying oxygen and carbon dioxide around the body. So they're two classic examples, the sperm cell and the red blood cell, of cells that you may like to choose when you're talking about structure and function. But there are a couple of cells which I'd like to mention, and I'm going to go into more detail with these because they work together to do the same job almost. We have a goblet cell and a ciliated cell. So down at the bottom here is a cross section of your trachea, so your windpipe, and these cells, goblet and ciliated cells, can be found inside the lining of your windpipe and also lungs as well, or bronchioles, shall I say, so all along your respiratory system. So these goblet cells and ciliated cells, that's where they are found. And this picture on the left hand side shows you the difference between the two, and they're found very, very close together, but they're working together to do the same function, but they do it complementary. So they do a different job, but they actually do that job in order to do the same function for us, which is they protect our respiratory system. Now here, this one in green, hopefully you might be able to see it. It looks a little bit about like a goblet. So if you put a stem on that, it looks like a wine glass, and we call that a goblet cell. And next to it, we can see on this level here that there are lots of little fine hairs coming out at the top, and they are ciliated cells. Because they're cilia, those hairs are called cilia. So these two specialized cells work together to protect our airways. And when I sort of write an assignment, I think it's really nice that you can find cells that would work together, because I think it would flow really nicely to explain one and then the other. Whereas if you've got the sperm cell and the red blood cell, there's not too much you can say that link the two together apart from the fact that they are specialized cells. Now, if you want to choose those, it's absolutely fine. You can still do a fantastic job explaining them, but I like to put myself in your shoes, and I like to see where we can take it. And so I've basically done this research task as though I was learning for the first time, so I can show you kind of the difference between a pass, a merit, and a distinction as we go along. Now, it's very easy to pass most things in the course because it's superficial. So really, when we're talking about function, I'm going to talk about them both separately as we go on, but the function really is to protect the airways, to protect the trachea or the lining of the trachea, and they do it in different ways. So let's have a look. If I'm going to explain or write this in assignment, then I came up with this little quote here from a particular source that was of a journal, and you can European Respiratory Journal. And what it said was mucociliary clearance describes the self-clearing mechanism of the trachea. And it depends on the physiochemical properties of the mucus and the cilia. Now, basically what that's saying is they work together. It's depending on mucus and it's depending on the cilia, because it's the goblet cells. There's my wine glass. That's a goblet cell, and that's a ciliated cell. There's the hairs on top. It's the mucus that this thing produces, and it's the movement that this thing produces that helps us get rid of harmful things that are in our trachea. So I thought that would be a nice quote to sort of introduce these two specialized cells that I would be talking about. And the term is mucociliary clearance, which is mucus done by the cilia. And as you can see, it depends on the physiochemical. So there's some physical properties and some chemical properties of these two cells that help us to protect our airways. So what I've got to try and do as a student is to try and really show excellent of these. This is what your assignment is graded on, or many of your assignments are graded on: understanding, depth, accuracy, structure, and use of diagrams. So let's have a look at the goblet cells first. And I came across this in another journal of the same type. I wanted to find out about the mucus first of all, because the goblet cells are what creates mucus. And that's a really lovely quote that I can use, or piece of information that I can use to support the fact that I'm saying that the goblet cells produce mucus because it protects and lubricates the surfaces. Excuse me. And part of the issue, or part of the function of that, is that the mucus traps dirt and particles in the windpipe or the trachea, ready to be brought up. So I thought that was nice that I could see some very sort of introductory quotes. Here's an actual slide of how they look in real life. You can see that this is the goblet cell, and next door to it would be a ciliated cell. So you can see they're lined all across the tissue, and you can see the cilia at the top, and all this gunk on top will be the mucus. And what will happen is the cilia will push the mucus towards your throat to be either swallowed or to go into your digestive system to be broken up. So I found this image in Marieb, actually, Elaine Marieb's book Human Anatomy and Physiology, and I noticed at the bottom there, I mean, this was talking about a goblet cell. Didn't really say much in the text, but it says, notice the secretory vesicles and well-developed rough endoplasmic reticulum and Golgi apparatus, which got me thinking, why is that? So why has it got excellent Golgi, excellent rough endoplasmic reticulum, and loads of vesicles? So we can see in the diagram there's thousands and thousands of vesicles. Now that's got, well, it says it's containing mucin. not mucus, but mucin. Interesting. I'm not sure what that is. So I thought, let's find out about that. What is mucin? So I'll put a question mark on that. And then you can see down here in this middle section is Golgi bodies and rough endoplasmic reticulum, which is going to be producing these vesicles. You can see it. It's what we've been doing in our generalized cell. That's what packages those proteins into the vesicle. And at the bottom you've got the nucleus, which is giving the codes to make the proteins. So I thought that was a really useful image. Although it doesn't say too much, I can apply my knowledge and think, well, actually what's going on is that the nucleus is giving the DNA copies, the rough endoplasmic reticulum's making the proteins, and the proteins are getting packaged in these vesicles ready to go when we need it. Now, when we say ready to go and we need it, I also thought, well, what triggers the release of this mucus? Are we producing it all the time? And I found that in this aspect that mucus is discharged in response to a variety of stimuli, and it can be discharged in fractions of a second. So whenever the airways get rubbed up the wrong way, whenever they get offended, then we can just spill out all this mucus into our linings to help protect it. And another thing that it says is that they can increase in number. So actually the goblet cells can go through mitosis and make sure there are more goblet cells on the lining of the trachea, which is why maybe some people produce more mucus than others. And then it comes back to the question that I set earlier in that what is mucin? And I thought, well, what is mucus? What is, what's in it? And so I came out with that mucus has a property or a chemical in it called lysozyme, which is a protein. It says there it's a protein. And so I thought, right, well, let's have a look at what a lysozyme is. And lysozyme, although this is very, very sort of hard going, I picked out that it leads to the degradation of peptidoglycan in the bacterial cell wall. So basically the lysozyme in the mucus breaks down the bonds in the cell wall of bacteria, which is bactericide. So it'll basically help destroy the bacteria that sticks to this mucus. Another structural feature is that I picked up on for this diagram is that it's got microvilli on top. And I'm just wondering what do we think the microvilli or villi perform? What role does it perform in terms of producing or discharging this mucus into the trachea? And really, microvilli are there to increase surface area. Again, it's surface area. So if we measure this flat membrane, it might be 5 centimeters long. Whereas if we put folds in it and we measure that, it might be 15 centimeters long. But if we put little dimples on each one of those folds, it might be 50 centimeters long. And so basically in the same distance from here to here, we've actually got in 10 times as much membrane as we did with just a flat surface. And that's what the microvilli do. And you have to ask yourself the question, why is that good for a goblet cell? Well, because it can spew out far more molecules of mucin or mucus than if it was just a flat barrier, and that's why goblet cells have microvilli. And here's a quote that supports that, so I could use that in my assignment beautifully to support the image that I found in a textbook. Goblet cells lack cilia and have occasional microvilli which increase the surface area for secretion. Perfect. So here's a summary of the goblet cell. You've only got a couple of hundred words for both specialized cells, but I hope you can see that actually if you do a bit of research into it and you don't just accept what people say on the face of it, you can actually delve deeper, and that'll help you to show understanding further along the line. I'll let you pause this at any time and you can have a read. I'm not going to go through it. So then we look at the ciliated cells. Well, how are they suited to their job? Well, ciliated cells, as I've said, beat their cilia across the lining of the trachea, and that'll take the mucus up towards the back of the throat. And I've got a nice introductory quote here. Its cilia continually propel debris-laden mucus towards the pharynx. So basically, if that's the trachea and there's the mouth, the cilia beat so the mucus goes up towards the back of the throat, and then we can either spit it out or we can swallow it. Cilia are hair-like structures made from microtubules, and again another quote just to say that they move fluid over the surface of the cell, and you can find almost 1 billion per centimetre square on your trachea. Now that's insane. It shows you how small they are. 1 billion per centimetre squared. And here we've got a cilia which basically beats, I suppose it's like almost seaweed in the sea when a tide washes over it. They beat in a power stroke, and if you can imagine there's one here, one here, one here, one here, one here, and they all beat in the same direction, then that thing is going to be pushed forward, or whatever's on top of it just gets pushed forward, and that's basically what happens. You can have a look. If you Google it, actually, if you YouTube it, you can see some cilia in action. And what that does, I found this term, the mucociliary escalator. So here's the mucus, these sun-shaped things are spilled out by the goblet cells, and it's the ciliated cells which push up all of the sticky stuff containing all the bacteria and the nasty stuff towards the pharynx so that we can get rid of it. On an earlier slide, it was written that the ciliated cells are part of the pseudostratified epithelium. You can go back if you don't believe me, but I'm pretty sure it does. So then I got to thinking, well, what does pseudostratified epithelium mean? And basically it means closely packed cells that make up the outer surface of the body. Right now, without me thinking about it, I would have normally read that, passed it through, but I didn't want to accept that as standard. I wanted to know, what does pseudostratified epithelium mean, and is it related to the structure and function of what a ciliated cell does? And so what I found was that it does. The pseudostratified, they're closely packed, as we've seen on the pictures before, and the fact is that they're on the outer surfaces of the body, i.e. they're on the inside of the trachea. So a little bit more of reading meant that I could found that ciliated cells, which are next to each other, like this, have something called tight junctions. So here's the cell, and here's the cilia all the way on top, which we've seen before. And the fact is that these cells are tied together almost by proteins. So let's do it in purple. These are proteins here that tie these cells together. And if we read on from that in terms of Marieb, sources at the bottom on page 66, you can see that bacteria, dust and other unwanted substances cannot lodge between the goblet cells and gain access to the bloodstream. So the fact is that nasty bacteria can't lodge between these two cells because the cells of the pseudostratified epithelium are squished together that tightly that nothing can infect it. And so it's a structure to function thing. Remember, the function is protection. How do they protect? Well, one of the reasons is that they're tightly knitted together by these tight junctions, which are these proteins that sort of stick cells together so that bacteria and other nasty things can't get access to the lovely bloodstream, which is the highways around our bodies. So that's another structure to function feature. So if I sort of just take a step backwards and just finish this off, In part 2 of your assignment, you have to explain the relationship between structure and function of two specialized cells. The way I recommend you go about it is to first of all describe the function of the cell that you're going to investigate, just the overall function, and then I would delve deeper to say why the structures allow that cell to do its job. So in summary, if I was talking about a goblet cell, for example, I'd be saying that the role of a goblet cell is to protect the trachea from harmful pathogens or bacteria or viruses. It does so by secreting mucin or mucus, which is a sticky substances that line the trachea and allows it to trap harmful pathogens. Now, if I've done that, I've kind of hit the criteria and I'm going to get sort of a passy kind of level. Then I need to go into that cell and think, well, why is that cell good at protecting the trachea? Well, the goblet cell is really good because it has excellent rough endoplasmic reticulum and Golgi bodies, which enables it to produce vast amounts of vesicles filled with mucin, which converts to mucus on the lining of the trachea. The fact that the lining or the secretion of the mucus is responsive to insult. So if the lining of the trachea is insulted by breathing in toxins or smoke, then those vesicles are ready to go via exocytosis and they line the trachea full with mucus. An added property of the mucus is that it's got lysozyme in it, which is an enzyme that breaks down the cell wall of bacteria. So it again provides a chemical property that stops infection and harm. Now, twinned with the ciliated cells beat their little hairs on the outside of the cell membranes, which move that mucus to the back of the throat where we can expel it, which is called mucociliary clearance. And you can go into that as much as what I've done on the slides. I'm not going to go through it all again, but I'm hoping this has been useful for you to gain a grip of how to relate structure to function in terms of specialized cells. The information is not easy sometimes. You can go as deep as you want. Now, if you're choosing the muscle cell or the neuron cell, you will have a limited knowledge maybe at this point in time, but that doesn't mean to say that you can't do it. It just might need a bit more of research. You're going to need to show resilience, so you might go down a dead end. You might even rip it up and choose a different cell to study. That's okay, it doesn't matter. What I would try and suggest is that you use a variety of sources to help you. So some online, some paper, some books, and just show that you've read around the subject, and that's going to give you more chance of getting a distinction because you've proven that you're an expert on that particular topic. And if you don't accept not understanding, that's going to really serve you well. So if you do read something like pseudostratified epithelium or lysozyme, and you just write it without actually not knowing what it means, then you get a much bigger chance of getting called out on that when somebody's coming to market, because you probably won't have explained it in your assignment. If you can explain it as you go, that's going to show you excellent understanding, and that's going to get you distinctions. So hopefully this video has been useful and as a guide. If don't worry about going for the erythrocyte or the sperm cell, that's perfectly fine, or any other cell, that would be also fine. You don't have to do the goblet cell and the ciliated cell. I just wanted to use that as an example. Hope this helps.